What Element believes very strongly in is that we aim to accelerate the learning process through collaborative discovery, meaning that the mysteries of biology are really only going to be understood collectively, and therefore we need contributions in technologies that span fairly broad areas of science. And that's been the spirit behind developing of the platform. Whether that's done in a more clinical setting, as shown in some of the pictures below, whether it be for rare and inherited disease, infectious disease, pediatric and adult oncology, and the like, are all areas that I've spent significant parts of my career prior to joining Element. And in fact, some of the projects that my laboratory has participated in are actually highlighted in the pictures above. Some of these were forensics. In the two cases of the children on the upper left and upper right, were rapid sequencing cases to find clinically meaningful diagnostics to improve the clinical care of those children. And then the two women you see in the middle is a fascinating case where they each carry a mutation in the same gene, two different mutations or between the women. But as you can see from their pictures, in one of the women, the mutation causes a fairly severe muscular dystrophy. But in the other woman, that mutation actually causes an ability to have a very unusual or very significant change in muscular density. And it's these types of variants and these types of changes and really understanding them at the genome scale is what's been a strong motivation for not only designing experiments and executing experiments across biology, but also helping to try to contribute to technologies that help open up more of biology. So if we focus on the sequencer, Element was founded, as I mentioned, 5 years ago, and the sequencer is just now being launched. And the foundational ask for that platform is shown here. Number one was to be a best-in-class data quality, and I'll show you some data of that shortly. A strong affordability in both the instrument as well as the reagents, and then flexibility in the workflow, the ability to have the ability to use a number of different options on the platform. So as we see here on the right-hand side of the slide, Element is shown with the broad green dot compared to a number of other platforms where we're plotting the price per gigabase in US dollars versus the output per flow cell in gigabases. And as you see from the plot and where it is, just one second, is this green dot is intentionally a little bit broad. So the specifications of the instrument are 800 million paired reads per flow cell. It's a 2 flow cell instrument. So this area of the instrument is where the 2 flow cells load. This top part of the instrument is a control screen, a touch control screen. This is one reagent nest for the right flow cell. This is the reagent nest for the left flow cell. And then behind this door is the waste bottle. So the 2 flow cells sit in here. They can be started asynchronously, meaning one flow cell can be started at time point A, and then at any time point in the future, the second flow cell can be started at different sequencing conditions if desired. The current kit is a 300 cycle kit, which enables paired 150 sequencing. And there's also enough reagents to perform up to 32 cycles of index sequencing. And so that may be in reading UMIs, index read 1, index read 2, et cetera. So it does support all manners of indexing. And the most common workflow is a paired 150 run with full indexing in a dual read indexing setting, allowing and supporting unique dual indexing or combinatorial indexing. And I'll mention some more of these features as we go through the presentation. But at the list price of the kit, which is $1,680 US, that gives us a per gigabase cost of approximately $7 at specification. But as you'll see in some of the data, most of our users are actually seeing around 1 billion to 1.3 billion reads per flow cell, although the specification remains at 800 million. We're seeing quite a bit higher than that, which that actually brings the per gigabase cost down in the $5, just over the $5 US range. We are launching a paired 75 kit later this year in Q4. that kit will cost $1,080 and is tailored more towards RNA-seq counting applications or single cell. And when used for single cell, at the approximate 1 billion read output, which we see in the vast majority of cases, that puts the pricing in the about $1 per million reads, which is highly competitive even with the NovaSeq S4 pricing on a per read basis at the paired 75 condition. So very, very cost effective. The reason the green dot is as broad as it is at the upper end of the dot is if at our specification price, and the lower end of the dot is our observed price in the field. So I just wanted to give some details around specifications and pricing. All of the specification details are clearly shown on our website as well, along with a series of application notes and a series of kits that we validated as compatible with the workflows on the instrument. Now, the next two slides focus on a couple of key points, and that includes the chemistry on the surface of the flow cell, as well as the sequencing chemistry. And I'll also mention what's shown on the lower middle and right-hand side of the slide in just a moment. So element surface chemistry is an entirely new surface chemistry in the sequencing field and was developed to have as low a background as possible under biological conditions, both sequencing as well as other conditions. And what you're seeing here is the proof of this low binding surface, which in the case of a higher background binding surface looks like this. If we take these same biological conditions and apply them to the element surface chemistry, you can see there's just this very faint outline of this very bright white dot. And this enables element to have a very high contrast to noise ratio, which of course is related to signal to noise. But we often refer to it as more important to have a high contrast to noise ratio. A good example of the difference between high signal to noise versus contrast to noise is thinking of observing stars in the nighttime sky, where the stars are not very bright, but they're easy to observe because the background behind them is very low, and therefore the contrast is high, versus during the daytime where those same stars are there, but they're just not observable because of the background light from the sun. And so that's maybe an analogy to understand element surface chemistry. So high contrast to noise ratio. Now, very interestingly, these other images you see here for single molecule antigen detection, as well as this cell detection, these were also captured on the sequencer, and both with the optical system as well as with the surface chemistry, so showing that it has a very extendable use possibilities. Now, this is certainly not to suggest that the platform has been designed specifically, or we're launching it as anything other than a DNA sequencer. What we're really showing is two things. One is that the surface chemistry and the optical system has quite a bit of opportunity for additional use in the future, because we want to give confidence that investment in the machine may pay dividends in other areas beyond sequencing over time. And the other is, as I mentioned earlier, Element is very committed to opening up biology to many different investigations. And so we look forward to working with researchers from around the world of coming up with new and novel applications for these surface chemistries and sequencing chemistries. But just we thought, and if I allow this movie to play, you can see that this antigen antibody interaction that's shown on the field was also, again, done in a cyclic manner, captured with the sequencer. So there's a variety of opportunities for us to move that forward. So I'm summarizing some highlights that I'll go into a little bit more detail on the right-hand side of the slide. And as I mentioned, from 2018, this is what some of our surface chemistry with DNA molecules looked like. And then through a rapid evolution process over the last couple of years, this is how the sequencing field of view looks as of 2020, and it's continued to be optimized. So we use a random flow cell. It's not a pattern flow cell. It is a 4-color imaging system, 1 color per base. Some of the clear areas for differentiation that we see is a very, very low optical duplication rate on the sequencer. These 4 different colored bars shown here are 4 different library types and 4 different applications on the Element platform. This is the comparison of one of those same libraries on a competing platform. the market leading platform as far as installed base, as well as one of our competitors shown here. This very low optical duplication rate is related to the amplification method that's used on the platform, which is a rolling circle amplification. So we do not generate high degrees of duplicate molecules, nor do we generate chimeric molecules, which allows index hopping to be negligible to non-existent. So our specification is an index hopping rate of less than 0.5%, which is shown with this red dotted line. But what we observe under the same 4 conditions that were shown here were also used to measure this. So this is 4 different library types with several replicates of each library type over different runs. You can see in reality, most of our index misassignment rates are less than 0.2%. And the reason for this misassignment rate is actually not due to the formation of chimeric molecules, but is actually due to image registration errors, which are very, very minor, but can occur. And we're continuing to upgrade and modify our algorithms to bring even these very minor levels down further. What's shown at the top is our quality score versus number of read pairs. So I mentioned the specification of the instrument is 800 million. And this is just a plot showing the effect of data quality as the instrument is overloaded. And so this gives that flexibility in that for the highest quality data, you can target between 900 million and 1 billion reads per flow cell and have as much as 97% Q30. But if you're performing counting applications or applications where the quality scores are not, or the highest quality scores are not necessarily required, and you're willing to sacrifice a small percentage of Q30 scores here, dropping down to 93 to 94% in this area, you can get about 40% more data out of the platform, sequencing at up to 1.3 billion reads. Or this also gives quite a bit of flexibility in the laboratory for loading concentrations and still achieving a successful run. And then what's shown in the neighboring plot is the same idea, but just on Q40 score rather than Q30 scores. And so our mean score on most of our runs is actually right at or slightly above a Q40 rather than Q30. So 1 error in 10,000 base pairs rather than 1 error in 1,000 base pairs. So a full order of magnitude better. And I'll show you some data in a few moments of where and how that difference in quality is best observed. The sequencing chemistry is summarized here on the left. And so I'll focus on this and some of the highlights. The sequencing chemistry is not a sequencing by synthesis method. It is a sequencing by avidity method, which think of avidity in terms of more like how antibodies bind in nature. Or stated differently, we separate the trapping step, which traps the avidite molecule to the homologous base that's next in line to allow the sequencing base to be detected and measured. And that's what's shown in this cartoon here, where we have an interaction of multiple nucleotides in what's known as a trapping step. After the detection, the avidite molecule moves away, and then the nucleotide strand is stepped forward by 1 base, and that's done with a stepping enzyme. So we actually have the stepping process and the trapping process are 2 completely different enzymes and 2 completely different steps, which allows us to have an optimal enzyme condition for both. Because we are not using fluorescently modified nucleotides, we can also use reagent concentrations that are about 10-fold less than competing platforms, which use a sequencing by synthesis method shown here. If you were to see our reagents and can contrast them to some others on the market, the reagent that holds our dye molecule is a very, very light shade. You can actually see through it. It's not a bright color, and that helps us keep our costs down quite low, as well as allows us to have a very strong signal to noise ratio as well. Because we do not use a modified nucleotide and don't have to cleave off a fluor, we also generate a scarless DNA molecule, which helps us control phasing and pre-phasing levels at a high degree of accuracy. So this is just proof of our Q40 scores. I'll show this in a little bit more detail in a moment. Where we see this in data on a human genome perspective is we have industry-leading indel calling. We see quite a bit, a strong improvement in accuracy in indel calling due to lower context errors in the data. And then a very uniform GC coverage, which is shown here. So what's highlighted in the red bars is 20% to 80% GC, and you can see it's very, very flat through these points over multiple runs. The instrument and algorithms, one of the other differentiating features is a very large field of view in the optics, as shown here. So it enables us to have very strong edge-to-edge performance. You can see the uniformity in these control flow cells on the dots. We developed, all the algorithms are developed in-house as far as the spot finding and this high density performance, which allows that flexibility out to the 1.3 billion reads. And then finally, the instrument also has a strong telemetry system to allow both remote monitoring of current and historical sequencing performance, as well as collection of telemetry data in an ongoing basis to allow historical runs and record keeping and even predictive analytics that allow us to observe when an instrument may need service proactively. Now, importantly, the field of genomics is not new and has been advancing for the last 12 to 15 years. And it was very important for Element that we recognize the creativity and the contributions and the innovations of so many researchers and companies that have come before us. And so we didn't want to draw a new starting line. We wanted to take advantage of the analysis methods, the library preparation methods, et cetera. And so that means we have two streamlined workflows to put samples onto the instrument. The first is an adept workflow, which allows any standard Illumina library from any vendor, as long as the P5 and P7 sequences are intact and it's a double-stranded DNA library. That kit can enter this adept library compatibility kit. There's no additional PCR, no additional amplification, and that circularizes the library and makes it compatible with the AVITI sequencer. So you can go directly from an existing library prep or your favorite library prep onto the sequencer with no other modifications other than the use of this kit. We are also releasing Elevate, what we call Elevate workflows, and that is what we have available today is you can substitute in our indexes and adapters with a favorite third-party kit to place libraries onto the instrument. And then in the future, we'll have true Element Elevate end-to-end kits that will take further advantage of some of the unique characteristics of the instrument. And we'll begin launching those kits in late Q3 to early Q4 this year. We see very strong indexing performance. I won't spend too much time on this. And then we see a typical indexing CV around 15%. You know, we did 4 brief beta tests, and you can see that our Q30 scores, our output per machine, et cetera, got better over time. These are the 4 beta sites that we performed early this year. First one was in late January. Last one was in mid-April. And then we've continued to advance the machine beyond even these times. All of the manufacturing for both instruments and reagents is all done in-house at Element in San Diego. And our site training prep is, we do that in advance. Installation of the machine typically takes 2 to 3 days, followed by another 2 to 3 days of application training. And we, of course, have built out a service and support team to support the instruments robustly. I did want to spend a little bit of time on the Q40 calling and scoring because I think it's quite important. The way we trained our Q tables was using 20 runs of human genomes. This was a PCR-free library prep, and that was to minimize any upstream errors. And that we used publicly available known data sets, and we used some of the genome in a bottle, et cetera, to make sure that we were we were accurately characterizing the variants that were observed. And then we were evaluated our training data using an additional 4 independent runs. And this is just some of the values for read 1 and read 2, the percent Q30 and percent Q40s we saw in these early training runs. I should note that these numbers, as we've made some chemistry improvements, have actually improved, and we continue to make these trainings. But I wanted to demonstrate that here is recalibrated Q scores versus predicted Q scores, with the solid line showing the predicted and then the dotted line showing the observed on the recalibrated Q scores in read 1 and read 2. So you can see very strong concordance between predicted and observed, and the vast majority of data being at and above Q40, as you see here. And again, this is a PCR-free library. I should note that all of this data is publicly available on the website, so anyone can repeat this analysis, as well as there's a number of other data sets available on the website for different applications. And if we do the same thing with a PCR plus library, you can see we can actually, the sequencing accuracy is high enough that we start detecting library preparation introduced errors. And we can see different levels of modulation off this curve depending on PCR amplification kits, et cetera. But you can see that effect that we see in this here. I should also note the recalibration of the Q scores is all done with open source software. This is the exact command line that was used. We try to be as transparent as possible with all of this training and example data that we have. We're more than happy to share these details with anyone interested in the future. I've already mentioned the coverage is a function of GC content. This is just seeing that a bit bigger, and then also proving that over multiple runs, we see this very significant range of very flat GC bias, allowing bacterial and other challenging genomes due to GC content to be sequenced very effectively with highly normalized coverage. I mentioned this a few moments ago again, that I just wanted to show it in a slightly bigger figure, that ability to flexibly load the instrument from above specification at 900 million all the way to 1.3 billion. And again, that effect on Q30 to Q40, very predictable and very consistent. Now, beyond standard sequencing protocols, we've also taken the opportunity to validate other applications on the platform, such as Olink Explore platform, and you're seeing the concordance between Illumina Illumina and element sequencing on the same sample. So very, very high concordance. That concordance is the same or slightly better than intra-run concordances, and allowing, again, the platform to directly use applications like Olink, where we go through the ADEPT protocol. We have done the same thing with 10x and some of their single cell work, as you see here. We do have a publicly available data set available for download from 10x on the website for anyone to look into this. But you see, very strong statistics above specifications for these applications, showing that the platform as it exists now is ready to use out of the box on a variety of applications. We've also had an opportunity to collaborate and look at conditions where we really wanted to answer the question, is the higher quality data we see on the platform, does it make a difference? And then what is, if you take advantage of the higher output of the flow cells, can you apply them in a meaningful way, say in a translational or clinically relevant manner? So in collaboration with the investigators shown on the slide at UCSD, we had the opportunity to look in a research-based project and at some consented samples that had a very rare eye condition. In summarizing this, what you can see here is using a single flow cell with just over 1 billion reads. We had Q30 scores at 94.6%, very strong index assignment rate, and a total yield of 326 gigabases. We intentionally sequenced the parents at a lower coverage to give higher coverage to the proband. And so here we have a 20x maternal, 21x paternal, and almost 52x coverage in the proband. If you add these up together, you actually notice this is over 90x total coverage, giving confidence that at this read depth and this yield of gigabase, we're able to actually generate 330x genomes per flow cell. And remember, the instrument is a 2 flow cell instrument, giving a high degree of flexibility. The turnaround time is approximately 44 hours, and that's whether you run 1 flow cell or 2 flow cells run within that 44-hour time window. And if you recall, the paired N150 kit has a cost of $1,680, and if we can achieve 330x genomes per flow cell, you're at less than $600 per genome in sequencing costs, without the need to pool large numbers of samples per run. So a very efficient capability. When we look at coverage per and total error, and this is looking at a genome in a bottle sample, HG005. And what's shown here in yellow is a publicly available Illumina data set, and then shown in red and blue is element data analyzed under 2 different models, and the specific analysis tools used here was Google Deep Variant, and this analysis was performed by Andrew Carroll at Google. And we're plotting total error so this is the total error for SNPs and indels, the sum of the false positive and false negative. And we can see at 15x, we have approximately 50% fewer errors in Illumina. At 20x, that number condenses some, and you notice that all of the platforms really have approximately the same amount of total error as we get to 30x, meaning that most of that error is taken care of by consensus sequence, which we've known for years is an advantage of short read consensus sequencing, as you can remove error as you go forward. What we're working hard to understand now is how we can lower this error to be as good at 20x coverage as we are at 30x, and we look forward to releasing some of this data and the path to achieving this in the near future. But we're working very hard to understand this. What we also want to really appreciate is, some of these differences here. Do they matter? And that's what we'll show here. So in this case, looking at HG005 again, 15x WGS, this analysis was also performed by Andrew Carroll at Google and on our behalf using our data, and was recently presented at the AGBT conference. So in this case, you're looking at heterozygous calls in red, homozygous calls here in yellow. You'll notice the 2 platforms for the calls that are made accurately are quite similar. But the difference is in the number of false candidates that you see at 15x coverage. Illumina shown here, large number of false candidates, and then a relatively small number of false candidates here in Element. Now, to be fair to Illumina, many of these false candidates will be filtered during the filtering of the VCF file. So that is a possibility, but it is still a significant amount of data in the raw calls. But if you recall the earlier chart, the expectation is this would collapse as we go to 30x coverage, but in fact, it does not collapse significantly. We still have this high degree of additional false candidates in the Illumina data compared to the Element. This is just showing us confidence that the Element data is just, by its nature, is much cleaner under these conditions. We can look at this over multiple runs and again see the same pattern, and including under different sequencing on Element, different platforms, PCR-free as well as PCR plus runs. And again, the trends are quite consistent when we look at that. We did have an interesting opportunity, and as if you recall the UCSD data where the parental samples were sequenced at lower coverage intentionally. So Google's Deep Trio, and which is described here in the bioRxiv paper, where the link is here, was designed to take advantage of trio-based sequencing to do more accurate and more specific de novo calling, which is very important in pediatric disease, as well as also a very good surrogate for somatic variation. And here's what we see in these results, which are quite compelling. So the true calls, and this is now using, again, a known truth set trio, the genome in a bottle set HG002, 3, and 4, but focusing only on chromosome 20. And that's just to make the data a workable amount of a very reasonable amount of data. So for true de novo calls on chromosome 20, there's just under 40, which is accurate. But if we ask how many false de novo calls do we make? And you notice that this is a significantly higher number. And the way this trends is although our sequencing costs are highly competitive, even with the most high output platforms, our sequencing quality and variant calling accuracy allows the downstream filtering and downstream annotation process to actually be quite a bit more streamlined, and therefore saving both costs as well as time in these downstream applications. So again, a fairly compelling set. I did also want to mention one last piece of data, and this is the last slide in the presentation, and that is we've also been looking at low diversity library sequencing and performance, and this is now looking specifically at methylation libraries, so bisulfite or enzymatically converted libraries. In this case, we're specifically using New England Biolab's enzymatic conversion. And so this is where we started, shown in this plot here, and this is again where we started, where we saw this drop off of Q30 at the end of the read. This is a Q score of 30 highlighted with the red line, and you can again see where we dropped off. So we knew that this was suboptimal in this data here. And notice that we This aligned rate is indicative of the percentage of phi X that was used in the run. So we were supplementing the run with 30% phi X, trying to maintain a high pass filter rate and high Q30 score rate. But note our pass filter rate was only 86%. So we developed a low diversity analysis pipeline, and then we're able to achieve a much, much improved performance where now our pass filter rate is at 96%, but we still had to include 30% phi X. And we knew that this, having to give away 30% of the reads was not optimal. So we reworked that, and we've now achieved the following capabilities. And this is all with, again, methylation libraries. So note the pass filter rate are all above 93% when we have either 0% phi X, 2% phi X targeted, or 5% phi X targeted. So we were a little bit over here, a little bit under on this one. But with only 2 to 5% phi X, or even 0% phi X, we can still maintain very high Q scores, very high pass filter rates, meaning that we now have it's an optional capability of using phi X in some of these low diversity conditions. And we're not stopping here. We're continuing to work on this quite hard. As you go up in library loading concentration and as you go up in density, and I should say this pass filter density translates to between 900 million and a billion reads, we do see a little bit higher sensitivity in these low diversity libraries to getting sequencing in the 1.2 to 1.3 billion reads. So we do know that if we push this out to the same degree I showed you in some earlier examples, there will be a slight loss in pass filter rate. So that's why one of the reasons we're continuing to work on this. So I know I've covered a large amount of information and large amount of data, but it was really in the effort to prove and show that although the platform has been recently commercially launched, it is quite mature relative to the competition and is ready to be used under a variety of conditions, and we can support all major library preparations. On our website, there's 13 application notes and then a large number of kits and other things that have been validated on the platform where we truly can support the vast majority of applications that are used in the field today. And anything that we do not support or isn't directly shown, we're delighted to hear from investigators so we can turn that into a priority. And so I'll just recognize all of the people at Element that have contributed to this, which has truly been a monumental team effort over the last 5 years, as shown here in a recent company photo. So thank you for the opportunity to present, and to be more than happy to answer any questions.